Good afternoon, everyone. My name is Emily Park, and today I'll be presenting you a paper titled Inhibition of Radiation-Induced DNA Repair and Pro-Survival Pathways Contribute to Vernostat-Mediated Radiosensitization of Pancreatic Cancer Cells. These are the author right here, and this paper will overall talk about what pancreatic cancer and Vernostat is, and how Vernostat mediates the radiosensitization of pancreatic cancer cells. So what is pancreatic cancer cells? As it is said in the name, location occurs in pancreas, which sits behind the stomach, and it has two types of cells. One is endocrine and other is exocrine. Endocrine cells secrete hormone into blood vessels, while exocrine cells secrete pancreatic enzyme into pancreatic duct. And depending on in which cell the tumor occurs, it will determine the types of pancreatic cancer. And the more common cancer type occurs in exocrine cells. Pancreatic cancer cell is very aggressive, and its symptoms is not specific in the early stages. Therefore, it is the fourth leading cause of cancer death in US. It is frequently incurable due to the fact that 2/3 of patients show metastasis, and it also contains inherent chemoradiosresistance. One explanation for inherent chemoradiosresistance is due to the fact that its prevalence to multiple redundant signaling pathways. This means that even if you block one pathway with the drug, it will still be able to signal other pathways, which allows the cell proliferation. Now that I talked about pancreatic cancer cell, what is Vernostat? Another name for Vernostat is suberyl-aniline hydroxamic acid, and it is a type of histone deacetylase inhibitors. Histone deacetylase inhibitors inhibits histone deacetylase, which removes the acetyl group from the histone, which allows the chromatin to be condensed and repress the transcription of the tumor suppressor. And this will lead to proliferation of cancer cells. However, histone deacetylase inhibitor comes in and blocks the histone deacetylase and allows the genes to be expressed and to tumor suppressor and inhibit the cancer cell proliferation. And Vernostat contains cytotoxicity, such as cell cycle arrest, apoptosis, and differentiation. And it is also known that Vernostat is synergetically able to work with radiation therapy to produce anti-cancer effects. And knowing all this, the researchers of this paper came up with a hypothesis. Combination of Vernostat with radiation therapy may enhance the radiosensitivity of pancreatic cancer and improve efficacy of treatment. And they looked at 3 possible mechanisms that Vernostat could use to radiosensitize the pancreatic cancer. And in this presentation, I'll be focusing on the apoptosis. So before we get started on how the research was done, they first had to obtain the materials. They obtained 3 types of the pancreatic cancer cell lines. One was called MIA-PICA2, another ASPC1, and COL257FG. Then they obtained Vernostat, which is shown in the picture here, from Aton Pharma, and they also had obtained dimethyl sulfoxide, also known as DMSO, to use as a control group. First, they wanted to confirm that Vernostat and radiation reduce the cell proliferation. So they used a clonogenic cell survival kit. What they did here is the cancer cell lines were treated with DMSO or Vernostat, which allowed the researchers to compare between the control group and the test group. After the treatment with Vernostat, they treated with radiation, with cesium. And after the radiation, cells were washed and trypsinized. The purpose of trypsinization is to unbind these cells from the medium so that it can be incubated in a different medium for colony formation. And once the colony were formed, they were stained and measured so that they can count how much, measure how much cells are able to proliferate against these radiation and vorinostat. So the results show that pre-treatment of vorinostat before the radiation repressed the survival and proliferation of the cell. As shown here in these all three diagrams, which shows the three different cell lines. The line indicates the control group, and dotted lines indicate the cell lines that were tested with vorinostat. And as it shows, the surviving fraction decreased significantly compared to the control group. And among these, the BIPACA2 displayed the maximum resistance. Next, they wanted to analyze whether apoptosis is a mechanism that vorinostat used to radiosensitize the pancreatic cancer cell. So to do this, they used immunoblotting analysis. And to do immunoblotting analysis, they had to use SDA's page of the whole cell lysate to separate all the proteins within the cell. And the proteins that were separated were then were in the gel, and the gel was put in between the negative membrane and the positive nitrocellulose membrane. And once the power was applied, the protein transferred from the gel to the positive membrane. And right here, this is the membrane. They added a primary antibody that detects the specific proteins. Then they apply secondary antibody, which contains enzymes that will bind substrate, and this enzyme will cleave the substrate to produce some type of staining, which will stain the proteins they are attached to. And once that was done, they were able to visualize the proteins. And in this experiment, they only used MIA-PACA2 cells because, as I mentioned earlier, MIA-PACA2 cells contains the maximum resistance against vorinostat and radiation. And the key apoptotic proteins assessed here is caspase-9, caspase-3, and PARP. The top here is a caspase-3. Caspase-3, if you see the far right column, it shows that vorinostat and radiation combined induced cleavage of the apoptotic proteins, which means apoptosis occurred. However, it also showed that on the second column, vorinostat alone induced cleavage. And if you see the third column, the radiation alone had no effect on the cleavage of these apoptotic proteins. And the same thing occurred in the PARP apoptotic protein. More cleavage has occurred in the vorinostat alone, and on the radiation, there was not much of a cleavage. However, the results were a little different in caspase-9. It actually shows that the caspase-9 were cleaved even in the control group, which means that it probably gets cleaved even in the regular cells, even in cells without any treatment with it. So what can we conclude from this result? I have mentioned earlier that pancreatic cancer cells have inherent resistance to chemoradiation therapy, and that is mediated by prevalence of multiple oncogenic signaling pathways. So because we know that just one treatment won't be able to block this multiple oncogenic signaling pathway, the researchers has combined two treatments, vorinostat and radiation, to see efficiency of enhancing the anti-cancer effect. And they have found out that the vorinostat enhanced the efficacy of radiation therapy. However, it was not through apoptosis. Then what other mechanisms does vorinostat use to enhance the radiosensitization of pancreatic cancer cell? If you read further into this research paper, it does talk about that inhibition of vorinostat uses the mechanism of inhibition of DNA repair inhibition of EGFR and NF-κB signaling to actually radiosensitize the pancreatic cancer cells. And to go further into the research, they will need to find more the molecular mechanism underlying these effects, because right now we know that it will radiosensitize, but we do not know that with the mechanism behind it. And this is a reference. Thank you.